webinar. And let me introduce the first speaker, Jean-Jacques Toulme from NOVAP, with its speech, Innovative Biosensor for the Detection of Influenza Viruses, a Sentinel Device. Please, you can share the screen. Okay. Okay, we can see it.
Do you hear me?
Yes, yes.
Okay, perfect. Good afternoon, everyone. So I will describe what we did up to now in the frame of e-farm. Our contribution relates to the engineering of biosensor for the detection of viruses in air samples. And these sensors are based on aptamers. So I will quickly introduce, I cannot move the slide. Good. That's interesting. Okay, it works now. So I will quickly introduce the aptamers and the method by which we get such aptamers. And I will say a few words about the antigens that we actually target for the detection of the virus, meaning hemagglutinin and neuraminidase, and how we did select aptamers to hemagglutinin before their characterization and their inclusion into an apta sensor. So very quickly, we start from a randomized library of oligonucleotides that contains something like 10 to the 15 different sequences. And we put this mixture of oligonucleotides in front of the target. We collect what is associated to the target, and we get rid of all the other oligonucleotides. We pick the few candidates that were associated to the target, and thanks to the parts are fixed regions that are identical for any oligonucleotide in the mixture, we perform amplification by PCR in order to generate an enriched pool following the first step of enrichment. And we put this first generation of oligonucleotide in front of the target one more time. And we perform this way, alternatively, selection and amplification under conditions that are more and more stringent, meaning that we are trying to drive the evolution of the population toward candidates that display increased affinity for the target. When this selection is completed, if we are lucky, we get aptamers. Actually, aptamers are interesting ligands. They display binding properties equivalent to that that are displayed by antibodies with a number of advantages. So I will not detail all the advantages, but importantly enough, I would like to stress that these aptamers are obtained by an in vitro process. So we don't use cells or animals, no live material. And this way we can get ligands that display a binding constant in the micromolar to picomolar range, depending on the size and on the properties of the target molecule. These molecules are obtained by a chemical process. We can introduce defined modification in order to get tools that will possess, that will display particular properties for the recognition or for the detection. And thanks to the chemical synthesis, we can get a large amount of oligonucleotides, a large amount of candidates. This way, there is no limitation regarding the sourcing and the reproducibility from batch to batch. And Last but not least, the production of these oligonucleotides is rather cheaper than the cost of antibodies. So this is a very long process when we have to run from this library that contains something like 10 to the 15 different sequences to a tool that will be adapted to a function. In this case, the detection of the influenza virus. So at first step, as I mentioned, we get an enriched pool, let's say, that contains something like 10 million different sequences. And we analyze these pools by bioinformatics. We try to identify characteristics either at the primary sequence level or the predicted secondary structure level. And we analyze something like 100 candidates. And we synthesize these oligonucleotides and we evaluate their binding properties. And at the end, we get aptamer. This is not the end of the story, because we have then to optimize these aptamers, meaning most of the time we reduce the size in order to get something that can be synthesized more easily and for reduced cost. And after that, we get the function. So we conjugate, for instance, this candidate to some dye for fluorescence or to some other function for immobilization and support. And at the very end, we have a tool. So this process involves a number of expertise in molecular biology, bioinformatics, molecular biophysics, and nucleic acid chemistry. So our goal is to obtain aptamers that can be included in a sensor for the detection of the avian influenza virus. Influenza viruses circulate worldwide in human beings, in different mammals, and in birds, and they are spread through respiratory droplets. This virus is responsible for huge economic losses in farms, especially for poultry. And given that the virus can eventually infect human beings, this is a threat to public health. So several avian influenza virus actually have infected humans. This is a limited number of cases, but nevertheless, this is something that we have to keep in mind. So the two major antigens on the virus are these two proteins, hemagglutinins and neuraminidase. So they are outside the envelope of the virus. They point outward, and they are recognized by different elements. Firstly, this is the way for the virus to infect the cells. And this is also these epitopes that are recognized by antibodies. So we have a large number of variation of these different antigens. And this is why the viruses are characterized by a number for the hemagglutinins and for the neuraminidase. So you certainly are aware of these, for instance, H5N1 virus that is responsible for the disease recently. So we picked the hemagglutinins, recombinant hemagglutinins. That was a full-length protein, almost full-length protein. This was H5, hemagglutinins 5, and we performed several selections of DNA aptamers against this hemagglutinins. These selections were performed either manually or on an automated platform, and we did run the selection over 10 to 15 different rounds, starting from different libraries with various characteristics regarding the size of the windows, the stringency for driving the evolution of the pools. And once we have performed this selection, as I mentioned before, we get an enriched pool that then will be analyzed for identification of potential candidates, essentially by bioinformatics. So we picked, following this analysis, this bioinformatics analysis, we picked, let's say, something like 100 different candidates. And we performed the chemical synthesis of these candidates that we analyzed in order to make sure that they are pure enough and have the expected size. And then we evaluate the binding properties of these different candidates by two biophysical methods, surface plasmon resonance of biolayer interferometry. From this analysis, we are able to evaluate the binding constant and the rate constant for association and dissociation. So very quickly, what we do is to immobilize the candidate on a sensor or on a chip, and we flow the partner, in this case, the hemagglutinin, over the chip. And when there is interaction, we observe an increase of the signal that corresponds to the association of the hemagglutinin to the oligonucleotide. So in this case, this is the resonance signal. When we stop the injection of hemagglutinin and we introduce the buffer, progressively the complex that was formed between the oligonucleotide and the hemagglutinin start dissociate. And from the analysis of these two different curves, we are able to get access to the binding constant. This is what we call a sensorgram. So we did perform this characterization by SPR or PLI on the different candidates that we synthesized. And here on this sensorgram is shown the results of a few of these candidates. So you see that in gray, this is the negative control, so library that does not bind to hemagglutinin. And some of the candidates do show a nice association signal, and then dissociation that is rather rapid for this particular candidate and slow for this one and this one. So in these two cases, the association is extremely stable. And for these three different aptamers that are listed here, we get a KD binding constant in the low nanomolar range. And we checked that these oligonucleotides were specific for hemagglutinin. So as I said before, in order to play easily with these aptamers, we try to optimize them. And the first thing we do is to truncate and restrict the oligonucleotide to the part of the sequence that is requested for the specific recognition. So we start from something that is likely like 80 nucleotide long, and progressively we delete part of these oligonucleotide in order to minimize the size. And for this particular aptamer, for this particular aptamer, we were able to reach a size of 37 nucleotide. The binding constant that was evaluated by SPR in following the injection, the successive injection of several concentration of hemagglutinin was evaluated to be about 5 nanomolar, meaning that we were able, in this case, to improve the binding of this truncated version, this one, compared to the parent aptamer, the full-size aptamer. So now we can play with this optimized aptamer for engineering the sensor. Just a few words before the engineering of the sensor. I mentioned we have a number of variants for the hemagglutinin, 1 to 19, something like this. And we evaluated the binding of our aptamers to different variants of hemagglutinin coming from either avian virus or the swine virus or the human virus. And you see that some aptamer, for instance, is able to recognize several hemagglutinins, whereas some others are very selective and recognize only this one, H5 from the AIV. So once we have this optimized aptamer, we introduce it into a biosensor. A biosensor is a device that is composed of a biorecognition element that will be specific for the target. And this biorecognition element is associated to a transducer, something that will deliver a signal that is able to indicate that actually this element interacts with the target. In other words, that we are dealing with a sample that contains the target. Then the transducer sends a signal to the detector. So in our case, we play with aptamers as biorecognition element, and our goal is to recognize viruses. At the first step, we will detect hemagglutinin, and thanks to two different technologies, first we use an optical signal, fluorescence, and second, an electric signal, the GFET. So I will now describe these two different sensors. So for the first one that allows the detection by an optical signal, we take advantage of what we call a molecular beacon. In this case, the aptamer, this optimized aptamer, is associated to a complementary oligonucleotide. The complementary oligonucleotide is conjugated to a quencher that brings this group next to the fluorophore that is conjugated at the end of the aptamer. In this case, the proximity between the two molecules here, the two markers, leads to an extinction of the signal. When this design meets hemagglutinin, the binding of the hemagglutinin to the aptamer induces the release of the quencher strand. So now there is no longer proximity between the quencher and the fluorophore, and we get a signal. So we perform the analysis of these different combinations, and one example is shown here. And when we use these for titrating with the quencher, we see that we reach a situation under which we have a reduced signal compared to the free state of the oligonucleotide of the aptamer. And this signal is restored upon the addition of hemagglutinin. So we have a concentration-dependent signal that translates the presence of hemagglutinin in our sample. So another sensor that we are working on, and I will show you only preliminary results. We recently started this in collaboration with a startup in Grenoble, Rafael. So in this case, what we do is to immobilize the aptamer against the hemagglutinin on a graphene film. So this graphene film conducts electricity, and the signal is dependent on what is associated to the film. So we have a basal level when the film is functionalized with the aptamer alone, and upon interaction with hemagglutinin, we have an alteration, a modification of the electric signal. So what we see here is that the signal increases when the concentration of hemagglutinin increases. As I said, it's only preliminary results, but they are very promising, and we are able to detect hemagglutinin in the low nanomolar range. So we are close to the end of this presentation, and this is where we are presently. We are developing a portable sensor using a small spectrophlorimeter for detecting the signal from our molecular beacon. So in collaboration with a startup company in Bordeaux, Goya Lab, we developed a small spectrophlorimeter, and this is able to make measurement of fluorescence for the detection of hemagglutinin. And on the other end, as I just mentioned, we are developing with Rafael this sensor that works like a lateral flow assay. So you just load a drop of a sample here, and you get an electric signal that translates the presence of hemagglutinin. And of course, the goal is to analyze not hemagglutinin, but the virus. So presently, we are working on the detection of the virus, and this has to be done in air samples. The sample will be connected thanks to the instrument that is developed by Counterfog. So in conclusion, what I showed you is that we were able to develop aptamers against hemagglutinin. I did not tell you anything about that, but we also have aptamers against neuraminidase. This is exactly the same situation. We have different neuraminidases depending on the virus strain, and we can combine the detection of neuraminidase and hemagglutinin for the characterization of the avian influenza virus. So these aptamers have very good binding properties. They display KD in the low nanomolar range. And now we are developing sensors, apta sensors, taking advantage of fluorescence emission and field effect transistor signal for the detection of the virus. And this opens the way to sensors that could be used on field by non-trained people. The goal is to have these measurements carried out by non-specialized technicians, and the results will be obtained in real time thanks to the transmission of the signal to a cell phone. This is true for the Goya Lab spectrophotometer and for the GFET apta sensor. The method for measuring is very simple, and the test will be a very cheap one. So I thank you for your attention. Here you have the people that have contributed to the development of this work, and we performed this kind of evaluation and assays on viruses thanks to a collaboration with Antonio Alcami and Angela Vasquez Calvo. And this was carried out under the umbrella of the eFarm project coordinated by José Luis Pérez Diaz. If you have questions.
Yes, thank you for your presentation. There is a question in the chat. And why GFET sensors have been preferred than lateral flow assays? What is the sensitivity of these sensors?
So the expected sensitivity is much, much higher. So with lateral flow, what you get is a colorimetric signal. So you analyze the density of a band. In this case, we can expect sensitivity that is much, much higher. And we hope to be able to detect in the picomolar range. In addition, this is really a quantitative signal. So there are many advantages to play with GFET compared to lateral flow.
Thank you for your answer. And our next speaker is Rafael Blasco. He will present new serological techniques.
Yeah, hi. Can I allow? Yeah, all right. I'll share my presentation. All right, can you see it now?
Yes, we can see the presentation.
The presentation. All right. So I'm going to tell you briefly about what we've been doing in the HeFarm project regarding the development of new serological techniques that are envisioned to help to characterize the situation in the farms. just to introduce you to the topic, when an animal gets an infection, usually there are several phases. In the acute phase, which lasts from days to weeks, we have a rise of the antigen and a rise also of the clinical signs of the disease. And later on here you see, I don't know if I can, okay, if I can get my pointer. Here you see that there is an increase in antibodies, which are typically of a class called IgM. And then later on, when the recovery starts, you can see a rise of another antibodies during the convalescent, the last until the convalescent of the disease, which are typically of the IgG class. So serology really compared to detection of the antigen that Jean-Jacques was talking about is a later phase in the disease, but it lasts much more. So it can give us the humoral immune response, which is a measure of the antibodies in the long term, and this is a useful technique when the pathogen is not easy to detect by itself, and also to see the immunological status of a population. I don't know why I'm not getting. Okay, there are several techniques to measure antibodies. Typically, the ELISA test, which is binding, detecting antibodies bound to a solid support. Immunofluorescence, which is a microscopic-based technique, and also Western blot, which I'm not going to describe in detail. So, in the context of HeFarm project, the project is based on the risk evaluation for the input and the exit of the pathogens, and I think this is centered about three things. The work in the project, which is first is based on the clinical science, which is a classical way of approaching diseases in farms. Then part of the project has been directed to detecting pathogens by metagenomics, PCRs, or otherwise, like aptamers or whatever. And then a third part would be to study by serology the status of the situation of the disease. And so the serology is really answering the question of what we have had rather than what do we have presently. Okay, the information is not a foolproof. I mean, a positive usually indicates prior exposure to a disease, but not only that, maybe also vaccination. And also negative results sometimes are related to problems. I mean, there are negatives, sometimes there are false negatives, but the results in any case have to be taken with caution. So I'm going to talk about also another technique, which is a virus neutralization test, where you can see the effect of antibodies on the infectivity of a virus. So usually what you do is you take your test antibody or serum and mix it with viruses, with a certain amount of virus, and then incubate with cells from some time, cells that you can add afterwards, and then you incubate. So you can see later on you have a readout regarding to measure the infection in that culture, which you can measure by cytopathic effect, by virus plaquing, or more recently is usually done by reported gene expression, like a GFP or luciferase reporter that can be read with the machine. And you can see here an example where a positive serum or antibody gives you a decrease in infectivity with increased concentrations, whereas usually you can distinguish that from a negative serum which does not affect infectivity. This technique usually requires the growth of the pathogen and usage of the pathogen, but it has several advantages, which is more. In principle, we can reduce all this to saying that it's more physiological than the other techniques. Well, this has been very useful with the SARS-CoV-2 during the pandemics recently, and it was very important to measure neutralization with antibodies because many of the variants of the virus were escape mutants that had acquired resistance to certain antibodies. So the problem here it was difficult to use the SARS-CoV-2, and it involved biosecurity in the labs. So it was in many labs we used other techniques which were like based on virus pseudotypes. In this case, we used the VSV pseudotypes, which is different, using a different virus, which is the VSV virus, and placing the glycoprotein from SARS-CoV-2 in the surface of this virus. This virus, this idea gets you to be able to measure the neutralizing antibodies. You can see here the comparison with the real SARS-CoV-2 neutralization test, and you can see that it really mirrors the normal situation. So we have, as I said, we have used the VSV system where we have a virus with a negative strand RNA genome, and we have substituted this the G protein, which is the gene that codes for the glycoprotein, external glycoprotein of VSV. We have changed it to a GFP that we can detect by fluorescence. So this is actually a replication incompetent VSV, which would make bold particles. And in this virus, we can add another glycoprotein that we get incorporated into the virus. So what we have done to begin with is to use vaccinia virus expressing different glycoproteins and do co-infection with VSV in such a way that when we infect with these particles, which are devoid of G glycoprotein, and we provide the vaccinia virus expressing a glycoprotein in trans, we can get these particles that have this foreign glycoprotein and which are single cycle infection. And this allows us to detect neutralizing antibodies. We've done this with many glycoproteins that we had expressed in the vaccinia virus system, and you can see here only that the ones that have the star were able to give us good titers after pseudotyping VSV. And I'm showing you this to show you that the situations are very different with different glycoproteins. So this was the first indication that it was not going to be a universal, easy system to go on. So our system consists of having from VSV, the VSV virus, a bold mutant, as I showed you, and then we decorate it with another glycoprotein, and we can do this with many different glycoproteins from different viruses. And in addition to that, we add a tag a molecular tag, which is a part of the genome that we can read by PCR and sequencing. And in this way, we have a mixture that where we can do binding with antiserum and separate the unbound viruses to the bound viruses. Then these virus pools would be read by doing RT-PCR of the mixture and then subjected to massive sequencing. Hopefully, this mixture and the readout of this mixture will give us binding activities to the antibodies. We have chosen two model viruses, swine influenza virus, because we have a collaborator who's got all the reagents, and also the porcine respiratory and reproductive syndrome, which we also collaborated with this woman from the Complutense University in Madrid. We've tried several different things with this setup, and we eventually, what we did is to express the head of the hemagglutinin, which is like a globular domain that we can separate from the rest. And the reason we did this is because expression of the whole glycoprotein was not successful. We had a lot of problems trying to get the complete protein to pseudotype. So what we did is to express this domain and fuse it to a part of other proteins so it would get incorporated. From this construct, we've made a version for the hemagglutinin 1 and another for the hemagglutinin 2. In both cases, these are swine influenza viruses. Also, similarly for the neuraminidase, which has a very nice globular domain as well, that we can isolate from the stalk and this area non-organized region of the protein. And we have, for this, we've done neuraminidase 1 and 2 as a test. And so with the idea with this and what we tried is to get the antibody, in this case an anti-HA antibody, and bind it to magnetic beads that then incubated with the VSV mixture. So once we have this, the beads are selected by a magnetic field and washed, and then of the bound VSVs in this pool, we can just go ahead and do the RT-PCR and the sequencing. And you can see here we have done several tests regarding the amount of sample we need, and you can see here that with 1 or 2 microliters is enough to get a good response for a positive serum. Just to give you a flavor of the results we got, you see here the mixture that we got with several of these, the H1, H3, and 1 and 2, and 2 antigens from peers, the porcine respiratory and reproductive syndrome, and you see that some sera are clearly positive to some of them. I think that this is tested in a mixture, so it's the relative activities is very clear. Some other sera are positive, but with a low amount. Some for the hemagglutinin and neuraminidase. So basically, to make it short, to go to the point, we got very good recognition in general of the hemagglutinin and neuraminidase, but we were unable to get any specific signal with the Pierce virus. So this was unexpected. So to conclude, we have set up and optimized this virus pseudotype system with the VSV. which took us a long time. And we tried constructs with many glycoproteins. As I showed you, some of them were viable and some others not. But unfortunately, the ones with swine influenza that we tried were not viable in the sense that we were not getting enough infectivity from the cultures expressing that glycoprotein. So we went ahead and did selected domains fused to the VSV glycoprotein, and this was more doable and more successful. And these fusions with domains of HA and neuraminidase were clearly recognized by positive sera. Well, this is more or less a proof of principle, and the good thing about this system is that you can do it in multiplex, and the readout is massive sequencing, so the possibility of expanding is enormous, and therefore, by making new constructs, it could be expanded to new antigens and disease. And I want to finish just mentioning the two people who did a lot of most of this work, which are Mia Figueroa here and Juana Sanchez Puig in the lab. And I'm going to leave it there.
Thank you for your presentation. If there are not questions, we can move to our next speakers, that are Sylvie Manguin and Clarisse Girault, and they talk about effectiveness of repellents for mosquitoes and ticks. I don't know if there is the... Okay. So there is some problem with the microphone of the speakers.
Do you hear me?
Yes.
Okay, nice. So it's good. It's working.
Perfect.
So I'm trying to project my computer. Good.
Okay. We are seeing the presentation.
Okay, nice. So hello, I'm Clarisse Girault, and today I will present you the results that we have obtained concerning the effectiveness of the repellent against mosquitoes and ticks. It was a work that was done by me and by two master degree students, Maximilien Barbier and Sutatlo Sotto. So this study was done to test plant-based environmental friendly repellents against arthropod vectors of pathogens. It was done under two research projects. The first one is the HE Farm project, and the second one is the CVEC project that was financed by the Pierre Fabre Foundation with a consortium from Europe and Asia. The objective of our study was to test the efficacy of 5 plant-based extracts
It seems that there are some problems with the connection. Maybe we can wait some minutes.
Insecticide with a low risk of resistance development, contrary to chemical products, and it has low toxicity and is environmental friendly. Concerning the material and methods that we have used, there were, for the plant extracts that we have tested, they were coming from Asia, and they have been chosen from several sources, local markets, healers' knowledge, and obviously scientific literature. And we have decided to select 4 Cambodian plants: Amomum cravan, Iptis faverans, Cinnamomum cambodianum, and Impatiens bocorensis, and 1 mix of Thai plants, the YY mix, which is a mix of ylang-ylang, guava leaves, and vetiver essential oil. Concerning the extraction of the plants, 2 methods of extraction were used for the Cambodian plants, either hydrodistillation for essential oil from the fruit of for Amomum cravan, or the bark of the tree for Cinnamomum cambodianum, or we used a maceration in hexane for the extract of higher parts from Impatiens bocorensis and Iptis faverans. Concerning the mosquito species tested, we have tested, like I told you, on Aedes albopictus, which is the most invasive mosquito species recorded on all continents except Antarctica, because it's well adapted to temperate and tropical climates. And their eggs are resistant to desiccation during several months. It's a mosquito that is mostly active during the daytime and can be vector of pathogenic agents, especially viruses, that touch mostly humans. Concerning the second species, it's Culex cancevaciatus. It's an invasive mosquito species recorded in tropical and subtropical countries of all continents, here in brown on the map. They lay the egg in form of floating raft, and the larvae can be found in the water rich with organic matters such like sewage, ditches, or latrines. For the time activity, it's only night time, and it can be this time vector of pathogenic agents both for human and animals, such as West Nile, that can touch human, but also birds and horses, or Usutu virus and echinocephalitis, for example. We have decided to rear these mosquitoes at our lab. For this, we had 2 strains. Aedes albopictus was a strain collected in Montpellier in 2016. And concerning Culex cancevaciatus, it was a S-lab strain that was at the R&D since 1966. For the rearing condition, we have used the same temperature and humidity for both species. The only difference was the photoperiod. It's really important for Aedes albopictus to have a longer light period than Culex cancevaciatus, because we have a risk that the mosquito can go in diapause. That means for example, during 2 to 4 months, the egg will not hatch. Concerning the alimentation of the adults, it was really important for them to have everyday access to sweet juice, and 2 times per week to have a blood meal to lay some eggs. Concerning the impregnation of the paper with the plant extract, we have used 2.8 milliliter of solution per waterman paper. For each plant extract, we have tested 2 concentrations, 2.5 and 5%. Each product has been diluted in ethanol, so we use absolute ethanol as control. And after the impregnation of the paper, we had to wait 1 hour for it to dry. For each test, we have used 15 mosquitoes, females, per replicate that have not been eating since 24 hours. And we have tested 2 conditions, either non-contact condition and contact condition that I will show you after. So for each, we have tested 60 females per treatment and 60 females per control. At the bottom, you can see the exciter repellency chamber. So you have a metal test chamber, and inside with the paper impregnated, and an external receiving box where we can see the mosquito by the top of it, since we have a glass that is put on it. So the exciter repellency tests are made in 2 conditions. Like I told you, the non-contact condition to see the repellency effect of the product. To do this, the impregnated paper are put behind the metallic screen inside the chamber, and the mosquito are put inside, and we see if they go to the receiving chamber or not. And we have tested the contact condition to see the irritancy of each product. And this time, the impregnated paper was in front of the metallic screen. So we put the mosquito in the test chamber, and during 30 minutes, we collect every mosquito that go in the receiving chamber. Concerning the results that we have obtained, for the repellent test made on Aedes albopictus in no-contact condition, on the left you have the percentage of a mosquito that has escaped, and on the right side you have the percentage of knocked down mosquitoes. Mosquitoes that are knocked down are on the ground of the box. They can't fly, they can't move, things like that. So for the YY mix, we can see that we have a high rate escape with 60% at concentration of 2.5%, and mostly 50% for a concentration of 5%. We have also a high knockdown rate at 2.5%, around 25. So we can see that we have an important exciter repellent effect. Concerning Amomum cravan, we only have a significant response at 5% with an escape rate of 40%. But at 2.5 and 5%, we can see that we have a knockdown. Concerning Cinnamomum cambodianum and Iptis we have a significant effect, but moderate comparing to the other products. Concerning impatiens bocorensis, we don't have any escape or any knockdown that was obtained. Now, if we look at the repellent test on Aedes albopictus in contact condition, so for the irritancy of the products, we can see that for YY mix, we have similar results that in non-condition contact. So we have a strong irritant effect. Concerning Amomum cravan and impatiens bocorensis, we have significant but moderate effect this time. And for Cinnamomum cambodianum and Iptis repellents, we have no significant difference between the treatment and the control. Here you have the comparison of repellent and irritant effect in non-contact, so the one in discontinuous gray, and the plane for the contact condition on Culex fasciatus. So here this graph is a little bit different because you have the percentage of mosquito that is remaining in the chamber on this axis. So you can see that for YY mix at 5%, we have 60% of repellent and 60% of irritant efficacy on Culex fasciatus. Concerning Amomum cravan, this time we have no efficacy that was shown, neither repellent nor irritant effect. So to conclude, the YY mix demonstrates robust and reproducible results at 2.5 and 5% on both mosquito species in both conditions, so non-contact and contact. Concerning Amomum cravan, it only shows a repellent effect against Aedes albopictus, mostly at 5%. For the three other products, they show less or no effect than comparing to the YY mix or the Amomum cravan. In the future, YY mix and Amomum cravan should be further tested with additional concentration, for example, 1% concentration or 10% concentration. Lab tests need to be done on ticks. For this, we will need to use ticks that have been reared at the facility of the Institute Pasteur. We will have to test the toxicity and remnants to be evaluated before any release in field conditions. We will also have to test on mice, then test on animals from the field for evaluating the efficacy of the plant-based repellents that we have used to protect both livestock, humans, and the environment. Concerning the laboratory for raising a tick colony in Institute Pasteur, it was acquired and equipped. I would like to thank José Luis Pérez Diaz and his team to have allowed me to work on this project. Sotavirek Bori and his team to have given us plant-based products from Cambodia, and also Professor Sylvie Manguin and Dr. Sarah Bonnet that were my PhD supervisors. And I would also mostly like to thank Maximilien Barbier and Sutatla Osupto that helped me to do this work. Thank you.
Thank you for your presentation. There are some questions. If there are not questions, we are now reaching the end of this webinar. Thank you to all our speakers for their valuable contributions, and thank you all for attending and for your interest in the eFarm project. We hope you found this session useful and inspiring, and have a great day. Bye.